So the topic to be discussed today is clonal selection theory. Clonal selection theory, before starting this topic, I want to share a quotation of an eminent immunologist, Silverstein, who suggests the, that an apt general description of the acquired immune systems approach at defending the host organism from pathogens. It took nearly 100 years to define and verify a theory that accounted for the diversity of antibodies. And what Silverstein suggests that, like Darwin's theory of evolution, much of the species of the theory may have been outdated given scientific progress, although there is no reason to abandon the core principles. Rather, it emphasizes the need to embrace the continually updated version of the theory. So his work provides a high level of survey of the development and core principles of the clonal selection theory of acquired immunity. So if we go to study about the clonal selection theory, first the question will come to our mind that means what is the basis for clonal selection theory? It is that how are the foreign substances, the antigens, are recognized by the immune system of the host of our body. So one of the great enigmas confronting early immunologists was that determines the specificity of the immune response for a particular foreign material or antigen. The general term for any substance that elicits a specific immune response by B or T cells. Around 19th century, Baudet expanded the concept of immunity beyond infectious diseases, demonstrating that non-pathogenic substances such as red blood cells from the other species could also serve as an antigen. Then the work of Karl Landsteiner and those who followed him showed that injecting an animal with almost any non-self-organic chemicals could induce production of antibody that would specifically bind to the particular organic chemicals. So here from the development of theories of this particular topic came different theories as it has evolved, and the earliest conception of the selective theory dates to Paul Ehrlich in 1990s. In 19th century, in an attempt to explain the origin of serum antibody. So Ehrlich proposed that cells in the blood expressed a variety of receptors, which he called side chain receptors here, side chain theory, which he called the side chain receptors that could bind to the infectious agent and inactivate. So this theory is abandoned when Landsteiner explained antibodies could be formed against not only natural antigens but also against various synthetic chemicals like riboflavin, estradiol, L-thyroxines, etc. and some organic chemicals that are non-self origin. In 1930s and 40s, this theory was challenged by various instructional theories. And according to these theories, these are known as direct and indirect template theories, A particular antigen would serve as a template around which antibody would fold, sort of like an impression mold. And this antibody molecule would thereby assume a configuration complementary to that of the antigenic template. So this concept was first postulated by Brinell and Holt. Then it was modified by Alexander and Mard. So they redefined in 1940s that the antigen enters antibody forming cells and acts as a template so that antibodies are formed with complementary combining sites to antigen. But in 1940s, Linus Pauling uh, showed that specificity was determined by folding of the antibody to form tertiary structure fitting antigen molecule. And actually, Linus Pauling was a crystallographer who was trying to predict structures of several protein molecules. So his prediction from the derived X-ray crystallographic data, the tertiary structure fitting, they're changing, they're folding. The antibodies and proteins, they're folding to form tertiary structures fitting the antigen molecule. So selective theories resurfaced as a result of new experimental data. Though the insights of McFarlane, Barnett, Journey, and Temogel, this model was refined into a hypothesis that came to be known as here what Burnett has said in 1949, that a genocopy of antigenic determinant was incorporated in antibody producing cell genome and transmitted to the progeny cell. So later on, this hypothesis that came to be known as a clonal selection theory. But before that, Journey in 1955, he has done a remarkable work. Actually, he has suggested that millions globulin molecules are formed in embryonic life with full range of antigenic specificities, and these are the natural antibodies that are developing after the infection or after the invasion of the antigen. That is why he said that this is naturally producing antibodies, globulin molecules, and the antigen when enters, it combines with nearly matching organic molecules, which is known as antibody. So the concept of antibody came, and this move on the antibody producing cells, they get activated and produce same kind of antibody. Means it is a natural solution. But this theory was countered by Linus Pauling. And this hypothesis that came to be known as the clonal selection theory, this theory has been accepted by the modern immunologist. And according to this theory, an individual B cell or T cell, B lymphocyte or T lymphocyte, expresses many copies of a membrane receptor that is specific for a single distinct antigen. And this unique receptor specificity is determined in the lymphocyte before it expresses them to the antigen. And binding to the antigen to its specific receptor activates the cell, causing it to proliferate into a clone of dot cells that have the same receptor specificity as the parent cells. So, what McFarlane Burnett proposed, he got Nobel Prize for proposing this clonal selection theory, and it is a deceptively a simple algorithm process that describes the diversity of antibodies and is the foundation for the understanding of the acquired immune system of vertebrates. So the question will come of what clonal selection explains. So this particular theory explains how the cells of immune system will interact with a specific antibody and antigen that enters the body. And this model was widely accepted. Now, what are the postulations? There are four postulations. The first one is that each lymphocyte bears a single type of receptor with a unique specificity, that is by VDJ recombination. So receptor occupation is required for cell activation, and the clone cells derived from the activated lymphocytes will bear receptors of identical specificity as the parental cells. And the fourth one, the last one, is the forbidden cells will be deleted at an early stage. So we come to the cell-mediated immunity. So there are two different types of cells. One is a B cell, and the second one is the T cell. So there are two types of B cells. These are lymphocytes, respond specifically to antigen on foreign cells, cells infected by pathogens and toxins released by the pathogen. So only on that time they will respond, and they will produce antigen. So type of B cells, these are known as the plasma cells. They will produce antigen, antibody, a complex. They will form a complex with the antigen, and they have a antigen recognizing site. So they trigger production of antibody. Antibodies are Y-cept, and they have a specific receptor binding site. Second type of B cell is a memory B cell. Although we have two types of memory B cell, T and B. Both produce in response to antigens by clonal selection and survive long term as memory cells. And for the secondary exposure to the same antigen rapidly gives rise to a new clone of lymphocytes, producing, here you can see, producing a rapid and greater immunological responses. But here, these responses or production of memory B cell as well as the production of plasma cell will not take place only by interaction with the B cell with the antigen. Here, the helper T cell is also required for the clonal selection of the B cell. Now, how do the B cell produce antibodies? The B cell develop from the stem cell that are there in the bone marrow of the adult, and then they are coming to mature into the lymph or mature and migrate to the lymphoid organs, say lymph node or spleen. And when the B cell encounters, mature B cell encounters with an antigen, it recognizes and stimulated and divides into many clones of plasma cells which actively secrete antibodies. And each B cells produce antibodies that will recognize only one antigen at each time. So it's a specific clone. Now, when the antigen is interacting with antibody, we have two types of responses. One is known as the primary response, and the second one is known as the secondary response. So the first exposure to a foreign antigen, a lag phase occurs, in which, here you can see, in this picture you can see that no antibody is produced, but here no antibody is produced after the first infection with the antigen. And but activated B cells are differentiating into plasma cells. Here you can see they are differentiating into plasma B cells, and then the lag phase can be as short as 2 to 3 days here, but often it longers. It is longer, sometimes as long as weeks or months. And the amount of antibody produced is usually low. Antibody level declines here again, declines here to a point where it may be undetectable. The first antibody produced is mostly the IgM type, and it's in few cases in sometimes producing small amount of IgG. But in case of second infection, reinfection by the same antigen, secondary infection occurs, secondary response occurs, and second dose of the same antigen is given days or even years later. An accelerated immune response occurs. Here you can see this accelerated immune response with production of antibody, huge amount of antibody. Here the lag phase is usually very short, 3 to 4 days maximally, due to presence of the memory cell, and the amount of antibody produced rise to a high level. Here, high level, and the antibody level tends to remain high for a longer time. Here you can see for longer time. And the main type of antibody produced here is IgG, and sometimes in small amount IgM is produced. Now, how the antibody mature, improving the fit to the antigen. So secondary immune response is not only faster but also produces antibody with up to a 10,000-fold increase in binding specificity or binding affinity. So the second response is more rapid since a number of memory B cells were produced during the first exposure. So here they are not only responsible for a certain rising in antibody production in the concentration in the serum, but also they are improving the affinity of antigen, affinity against the antigen with antibody. So clonal selection theory of B cell is caused by the antigenic stimulation. So first you can see the antigen can stimulate a particular type of B cell, and then the cell will proliferate, and they will subsequently they will produce two type of cells, different clones of two type of cells, plasma cells as well as the B memory cell. Memory cell, B cell, plasma B cells will then enter into the bloodstream, into the circulation, and then it will secrete antibody. Now, what are the consequences of antibody binding? There are four different consequences. So binding of an antibody with an antigen inactivates the antigen in four different ways. First one is the neutralization by opsonization, agglutination, precipitation, and these three reactions, then bacteria or viruses, they will interact, the antibody will interact, and then this will enhance the phagocytic cells, the macrophages, and under phagocytosis process, this can be eliminated. The fourth one is known as complement fixation. Here, the foreign cell, they will means the bacteria, they will interact with the complement, and then it will lead to lysis, cell lysis by the cytotoxic T cell. Then cell-mediated immune response. So how the microorganism entered and what are the responses. So there are two different type of response: cell-mediated response and humoral response. The cells responsible here, the lymphocyte, macrophages, and NK cells. So no antibody is taking place here. So after entry of the microorganism, it may be identified by the macrophages and then phagocytosis, or else it can release it, it can remove it, or else it will identify the antigen presenting cells, and then it will be digested into the endosome into peptides. The peptides must possess some epitope. So the digested peptides then bind with the antigen. In a class of which is known as HLA or human leukocyte antigen, and then they will means this particular type of antigen or a complex is a group of related proteins that are encoded by the major histocompatibility complex. So the type 2 major histocompatibility complex present on the APC antigen presenting cells, and then they will come and interact with the T cell receptor. The T cell will recognize the antigen and then it will activate the T cells. So the activated T cells then proliferate and will enter into circulation. So activation of T cells can also happen by the interleukins released from the macrophages. There are four different types of T cells and their functions are mentioned here. So helper T cells, cytotoxic T cells, suppressor T cells, and memory T cells. So there are two types of helper T cell, Th1 and 2. IL-2 activates other T cells, and gamma interferons. It increases the phagocytic activities. And then here the other type of T cells, the Th2, interleukin 4 and 5, B cell activation, proliferation of plasma cells, antibodies by the plasma cell, etc. The cytotoxic T cells, which is the CD8 plus T cell, they releases cytotoxic substances, mostly the lysosomal enzymes, and destroys own body tissue affected by the foreign bodies. The other type is a suppressor T cell. So the regulatory T cells so suppresses the activity of both the type of T cells, means the helper T cells and as well as the cytotoxic T cells, and the last one is the memory T cells. And these type of cells never enters into circulation, so they remain in the tissue. So the last one is the humoral immune response. So after entry of the microorganism, it may be phagocytosed or else may be identified by the antigen presenting cells. Then this will be digested by the endosomes into different peptides. Now the digested peptides will bind with the antigen, with the antigen HLA, which is MHC2 specific antigen, human leukocyte antigen, and then it will bind with the B cell receptor, activates the B cells. Then the activated B cells will proliferate, and they will produce two different type of cells, memory cells as well as the plasma cells. The plasma cell will produce antibody. So B cell activation and proliferation can also take place by the interleukins released by the macrophages. So far whatever we have studied that clonal selection theory is actually predicting how the B cells and T cells are preparing clonal cells, and how the antibodies are producing specific towards the antigen and to against the antigen. So these selection theory, clonal selection theory, is actually showing how specific immunity is developed in our body. That's all.